We'll wait for 5 minutes so that if anyone else want to join, they can join. Okay. Or I guess I can start and people can join in between. I hope so. Uh, Good evening, everyone. My name is Himanshi Singh. I'm a PMR PhD candidate in BSB department at IIT Kanpur. So today we're going to discuss about the week 6 assignment of previous year for the course Animal Physiology, which is taken by Professor Manik Das. You guys can still enroll, I guess, till today, if I remember correctly, for the certification. I uh, highly recommend it, that if you're spending enough time on it, getting a certificate out of it will definitely boost your CV and also your learnings. So I hope that most of you have went through the week 6 lecture, and if you have gone through them, maybe completely or partially, you know that this week was completely dedicated to neuronal system. We are very concisely covering the neuronal system because it is one of the major system, and we should know, at least we should cover most of our bases to uh, be sure that we understand about our brain, right? Because brain is one of the major organ which allow us to do all the things, right? It is called the control center for a reason, because whatever we are doing, whatever, irrespective of is it logical, illogical, if we are awake or we are asleep, in every situation, your brain is functioning, whether you know this or not, right? Whether you are actively involving in an activity or you are not actively involved in an activity, but passively in the background, your brain is functioning, right? To make sure that your survival is uh, continued. So, for example, if we take example of this class itself, right? So you guys know that today we have a class. So your mind was, as soon as the clock was moving ahead for this particular lecture's time, you started preparing, right? You started preparing your body, your mind, that, oh, now you need to go into a class, you need to sit and we need to discuss things, right? So there were certain characteristics for it. For example, if you were traveling, you will try to minimize the noise in your surrounding. Or for example, if you're at home, you will try to be in a like better area of your house so that you can understand and you can attend the lectures. So all these are active uh, things which you are actively doing to take part in an action. And here the action was to attend the lecture. So everything which we do, it is guided by the signals are sent by the brain. Now, how brain knows when to send the signal, right? This is what we covered previous week. That sensory system, sensory organs, sensory neurons are a surface through which we sense the outer environment, and again internal things which are sensed by our system. Those are the things which tells our brain what to do next, right? Yeah, what is the next step? Or okay, if we have this situation, how to act in this particular situation. even when you are asleep, right? So there are certain when you are deep into deep sleep, some of us, maybe you guys know this, that we give sudden jolt, right, when we are getting into deep sleep or we are in deep sleep. So it's such a way of a brain saving us, right? So if you read into it, I haven't gone into too depth of this phenomena, but what I'm aware of, and I might be wrong, so don't take my words here, but yeah, it is a way of your brain assuring your survival, because if you're getting into deep sleep, chances are you are having very slow breathing style, right? Because you are very relaxed, you are very calm, you are in lowest PMI possible, lowest breathing rate possible, because you're completely relaxed. So it is way of a brain to assure that you are still active. Not most of us go through this, but there are certain individuals who face these things, that their body jolt and shakes vigorously, not continuously, but a sudden movement of the body occurs when we are getting into deep sleep or we are in deep sleep, right? So even when we are deep sleep, we have dreams, we have nightmares. It is all unconscious part of our mind, right, which we unfortunately do not know much about. And uh, our unconscious uh, our unconsciousness is actually generated from the conscious world. If you know, there are some people who get very bizarre dreams, very very bizarre dreams, right? They are not able to connect the dots, and they're completely random dreams. I don't know, at one point I was at some place, at the other point I was somewhere else. I was not able to see myself. These all are some bizarre dreams on the internet. You guys can go and search for it. So all this unconscious mind and unconscious world is created by a brain only, right? But from where the brain is getting input, that's the question, right? Our eyes are closed. When we are asleep, our eyes are closed, our ears are somewhat dampened, but though still we are actively listening to our surrounding. Our touch senses, for a while we are not able to feel when we are in deep sleep. In between sleep and when we are turning and tossing, we do feel our surrounding. But in deep sleep, when we are completely into it, we are not feeling most of it. So all these things let us question this, that why and how, when there are no inputs from the surrounding, how our brain is able to generate this unconscious world. Correct? So this unconscious world is generated from the inputs which we got during the daytime, when our sensory system and our sensory organs were at par excellent and we were completely active. So if you want to decode your dream, because most of the time you won't be remembering your dream, but if you do remember your dream somewhat to some extent, the thing is, you might find that there were stimuli and there were certain uh, catches in the real world when you were awake, when you were conscious, that those were the stimulus which your brain took, and later it was trying to process it, and which generated this bizarre dream or a nightmare, right? So most of the time you can do that, but unfortunately we don't remember our dreams, right? So but if you are able to remember, then most of the cues, if you actually sit and think about them, most of the cues were taken from the world during the daytime when you were awake. So what does this tell us? This tell us that a brain is working on two parts, right? Just like any uploading and downloading file format on a computer system, right? You upload and you download. You upload the information, you download the information. So when such things happen and the unconscious world is emerging, it is a brain's way of processing the information which we got during our daytime from the sensory systems. And these sensory systems can be either your seeing ability, your hearing ability, your feeling ability, right? Your other ways through which you sense your surrounding, you sense your environment. So our unconscious mind, or when we are asleep, this is what our brain is doing. Our brain is not dampened now, right? Certain areas of our brain are actually stimulated because they are processing this information throughout the day. Just imagine how much amount of information and stimuli we are being bombarded, right? We can't even count because we are so habitual about it that we don't count our interactions and senses and all those things as a information or as a stimuli. But our mind, for our mind, in 24/7 it is getting information and more data to work on. So when we sleep, these data are getting sorted in our mind, right? Whether to keep this, not to keep this, where to keep it, correct? So if any of you is familiar with how the computer system memory works, so in that we have RAM, right? We have memories. So in a computer memory, what we have designated locations for certain things. So whenever you save a document or you save a text or anything, when you saving means what? You are putting it on some place in the computer memory, that when you want to collect it again, you know from where to collect it. But what you are seeing on your computer screen, it is just the representation of it. But if you go to the memory itself, in the memory chip where exactly the information is stored, and you know the information is stored in the form of zeros and one, correct? So the exact location of that saved file on a memory chip, we cannot know that. We cannot control it. We cannot access it by going to that particular location and extracting it, at least to layman. But certain computer scientists and software engineers, they can actually access these memories, and they can send information to particular location, then extract information from there. But for majority of us, we don't know how computer memory works, right? Similarly, our brain functions in a very diverse way. So during the day, when we are getting all the stimuli, all the information, all the uh, data points, right, to work upon, our brain keeps certain areas of our brain in a dampened way. Dampened way, I mean that it is keeping them suppressed, because that the processing of that information is not needed right there. For example, right now, if you're attending a class and I'm speaking and I'm telling you things, right? So my mind is constantly working in making sure that you guys are able to understand, and hence I'm formulating sentences in a easy to understand way, correct? So my mind is continuously working in making these sentences. I don't have any script with me, right? I'm just speaking as I'm like going through the lecture, correct? Which means my brain is now actively working. On the other hand, there are n number of things which are happening in my surrounding, in my environment. Even that information is getting transmitted to my brain, but I'm not actively looking out for it. Why? Because my primary focus here is to take this lecture, take this session, correct? Hence, most of the power and energy right now in my mind is going towards taking this lecture or giving this lecture. I not yet okay with saying that I give lectures. uh, Things aside. So, in a way, we can say that though we have the information coming to our brain 24/7, our brain has formed a way in which we can focus on one particular task at a time, let the other information and accessory data points and stimuli temporarily be stored at some place, so that whenever we get time, we can sort it later, right? This is how we also prepare for our exams, right? We'll cover the major topics first. We'll keep certain topics which are not so important for the later part. So if we get time, we'll sort that, or we'll read those things. But what is the priority? The priority has to be the important topics. So our whole energy, our whole planning, our whole timetable, our whole schedule is revolved around only trying to cover most of the important topics, right? And when we have time, then we'll go to the less important topics, correct? Exactly same. That is how our brain also works when it is receiving signals and stimuli and all the information and data points. Correct? So focusing on one thing, letting other things store at certain place of our brain, allow the unconscious mind. So what is unconscious mind? The unconscious mind, which we can name or we can explain, it's very, very, very difficult. But just for the understanding of this particular session, this particular lecture, we can say that when we are asleep and we are not consciously focusing on certain tasks, we are not directly in connection with our external environment, we are in an uh, uh, unconscious situation, right? And here unconscious doesn't mean that you are getting faint and all. It's just that you are deep asleep. You don't have any active information about your surrounding. So during the unconscious uh, duration, or say sleep, when we are asleep, these random information which we receive throughout the day starts getting sorted, right? And here by sorted, I mean, we don't know what exactly is happening, but we had the information which we got throughout the day. Now our brain has overloaded with the information because we focused on one task. For us, it was one task, but for our brain, it was a huge amount of work, right? We have seen the neuronal connection, right? We have seen those YouTube videos which I have showed you, how the neurons are making connections, if you remember, right? So those are the connections which are constantly being formed when we're focusing on one task. Only those pathways were getting stronger. Those neurons were making more strong connections when I'm focusing on one task. But when the surrounding information is also coming, which I'm not actively looking into it, even those are triggering certain pathways in my mind to store the information, to have some information about the actions and the stimuli which are present. So when we are asleep, we are now not actively focusing on one single task. So we have stopped focusing. Now this area of our brain is suppressed. We are not actively thinking, we are not actively looking for focusing on one single task. The other area with respect to the first one, then what will happen? It will be unsuppressed, right? Earlier it was suppressed because we were focusing on one task. Now our focus system is dampened down, it is suppressed. In comparison to this, saying that a temporary information system, a temporary information location where the stimuli and data were stored, now they are stimulated, right? Now our mind will try join all those pathways via neuron. It will generate a network from all the things which we received throughout the day. It need not to be in a sensible way. That is why most of the dreams and nightmares are bizarre, right? They make zero sense, correct? Because those pathways, those information which you receive throughout the day are just haphazardly getting connected from one point to another point. It's a very, very random process, right? Because you are not providing it a streamline. It is happening spontaneously. So if things are happening spontaneously, you are not providing a path or a direction for your thought, what will happen? Any pathway will get attached to any pathway, resulting in a memory or say dream or nightmare, which makes zero sense to us. But as I mentioned before starting with this, if you actually sit and think about your dream or nightmare, chances are the stimuli, the things which you have seen in your dreams or when you were asleep, you got it during the day. For example, if you saw any red car in which a joker is driving a red car, it's a very weird scene, right? And you have zero connection with this thing. You don't own a red car, or you haven't been to a circus where you have seen a joker, right? So if you see this, so in a way, in that particular day, you might have seen a red car pass by, but without focusing or actively looking into it, you let it go from your eyesight, but that information was stored in your brain, correct? Or you might have heard someone talking about taking their daughter for a circus. So from circus, what is the first thing which gets clicked in our mind? It is jokers, right? Jokers are there, colorful, funny beings, all that stuff. So now these two information were stored in your mind, correct? So when you were asleep, the brain doesn't know that the car incident and the joker incident are two separate incidences, and they should not be connected, because in your active, when you were focusing, when you were conscious, when you were a conscious being, for you they were two separate incidences. But for brain, when this information was randomly stored, for it makes no uh, reason not to connect this information, because there is no active bothering for this particular pathway, that you are actively saying no, 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 the car is a separate incident, the joker incident is separate incident, you are not supposed to mix it, because it makes zero sense, it is not logical. Hence we are not doing that when we are unconscious, correct, or when we are sleep. That is why you have bizarre dreams, because any neuron, any pathway is connecting any stimuli information which you receive throughout the day. Most of the time this happens, but sometimes also that they actually make sense, right? Our dreams and nightmares actually make sense. If we are genuinely frightened by something and we are having repeated nightmares about it, so it's a continuous thinking of that incident, or that thing which made you scared. So it's a active process in this way. So you are going to sleep by thinking that you were scared from this, something happened in that particular day. So chances of getting the same nightmare, same fear feeling is enhanced, because while you are going to sleep, you are actively saying, okay, this was the incident which happened today, right? You are reinforcing those incidences again and again, you are replaying them again and again in your brain, letting your subconscious mind, right, letting your subconscious mind know that these pathways, these incidences were strong enough to keep track of it, to keep this in this order only, what you have experienced, correct? Hence, if you are going to sleep by saying that you were afraid, yeah, something happened, chances of you getting a similar thing increases when you are asleep, correct? Even at that point, your mind doesn't know that these two incidences are separate. That was a real incident which I felt, but this is not a real incident, it's just a dream or a thinking of my own mind, it's a creation of my own mind, but they don't know that, right? So in this way, we have given a direction of thought for a night. So our unconscious mind will play that pathway again and again when we are asleep, because it has a direction. On the other hand, for the prior case, when I said it will create any pathway, it will create any random bizarre uh, memory, dream, or nightmare, because there is no direction for that particular thought, correct? So that is why we have conscious and unconscious separation of what to say, living. So it is not a clear cut distinction, or it's not, I don't even think that it's a classified classification based criteria that you can actually classify us like this. But still, we know and we all have felt in one way or the other way, correct? So this is our brain's way of using minimal energy. Like, you can ask why can't we store all the stimuli, all the information, process them all at same time? Because our brain is apparently most powerful, right? Even a smartest computer is not able to replicate what a brain can do simultaneously, correct? So the most powerful organ, does it really need this segregation of events, that if it is focusing on one event, it needs to keep the other information to a local place or a temporary place so that it can go through them later? Why cannot we have all the things in parallel? If I'm talking to you right now, I should actively take part and know what is happening in my surrounding and still be able to deliver this lecture without getting distracted, right? But we are not capable of doing it, right? So why is this thing? We are powerful being, our brain is a powerful organ, and still we are not able to do parallel tasks. And here parallel tasks won't be the thing that's scrolling your phone and attending the lecture, right? Because when you are scrolling the phone, it still be counted as a passive activity. Here, active and passive doesn't mean the requirement of ATP or energy. It completely means how through mind you are doing all those things. Correct? So it's just a beginning of a lecture, and I wanted you guys to understand that we have these separations because of the energy consumption. The more you think, the more information you are processing, the more energy your brain needs. So how our brain has evolved, what they did is they made sure that most of the energy input is going to a task which is right now important, and remaining everything is being stored for later purposes. Correct? So that we can balance our energy, we can use our energy wisely. Correct? So with this brief discussion about our brain, we'll see a very interesting disease which is called Alzheimer's. And I guess most of you have heard about Alzheimer's, right? And if no, then you might have heard about dementia, correct? So these are the terminologies, especially dementia. Dementia is basically, if we say in Hindi, bhulne ka bimari, right? That you are not able to remember things. And this problem of not remembering things is a severe problem. Unlike our daily deeds, right? For example, we left key somewhere, we were not able to remember where we left our wallet. These are very small things, not like this. In dementia, people actually forget who they are, who their spouses, who their children are. They are not able to recognize anyone in their life, correct? So it's an extreme form of that. So dementia is a consequence of something which happened in the background, especially in the brain. A neurodegenerative disease, as the name suggests, a neurodegenerative disease, right? A neurodegenerative disease means that your neurons are gradually getting degenerated, correct? And Alzheimer's is one such disease, which is a progressive neurodegenerative disease. What do I mean by progressive neurodegenerative disease? By progressive by progressive neurodegenerative disease, I mean that with time, as you age, correct? As you age, the severity of the disease will increase. And in a while, we'll see why this is the case, correct? So a neurodegenerative disease clearly means that your neurons are getting degenerated, right? And degeneration means the way they were supposed to function, the way their structure was supposed to be, the way their threshold potential was supposed to be, nothing is working like a normal neuron. And there can be n number of reasons. For Alzheimer's, that reason is accumulation of a protein. That protein which forms the amyloid plaque. Amyloid is a membrane protein in the neuron membrane. We'll see shortly in the video what exactly are those, so that you guys can understand what Alzheimer's is. So those proteins get accumulated in the brain, leading to the degeneration of the tissue, and hence decreasing the brain size, leading to dementia, right? So in on the slide, you can clearly see that in the figure A, these are the amyloid plaques. This is how a plaque is basically a concentrated globular kind of a thing. It's like a patch which you can see through multiple techniques, right? Yeah, so this is the amyloid plaque. The B figure says that this plaque is due to the amyloid protein. And as the stage progress, as the age progress, the severity can easily be seen that how it is being like getting to the whole of the brain, correct? This bar is here for the severity. So as the stage progresses, you can see that the red color is getting dominant, indicating that there's more damage to the brain, correct? And this neurofibril tangles is another protein which is associated with the Alzheimer's. I guess we can go to the video so that you guys know this terminology, and it will further increase your understanding, and then I'll explain it. So let me know if you guys can hear it and see the video. Okay, not here. Okay, wait a minute. I'm having some trouble sharing. Here it is. I hope you guys can see it and hear it.
Dementia isn't technically a disease, but more of a way to describe a set of symptoms like poor memory and difficulty learning new information, which can make it really hard to function independently. Usually, dementia is caused by some sort of damage to the cells in the brain, which can be caused by a variety of diseases. Alzheimer's disease, now referred to as Alzheimer's disease, is the most common cause of dementia. Alzheimer's disease is considered a neurodegenerative disease, meaning it causes the degeneration or loss of neurons in the brain, particularly in the cortex. This, as you might expect, leads to the symptoms characteristic of dementia. Although the cause of Alzheimer's disease isn't completely understood, two major players that are often cited in its progression are plaques and tangles. Alright, so here we've got this cell membrane of a neuron in the brain. In the membrane, you've got this molecule called amyloid precursor protein, or APP. One end of this guy's in the cell, and the other end's outside the cell. It's thought that this guy helps the neuron grow and repair itself after an injury. Since APP is a protein, just like other proteins, it gets used and over time it gets broken down and recycled. Normally, it gets chopped up by an enzyme called alpha secretase and its buddy, gamma secretase. This chopped up peptide's soluble and goes away, and everything's all good. If another enzyme, beta secretase, teams up with gamma secretase instead, then we've got a problem. And this leftover fragment isn't soluble and creates a monomer called amyloid beta. These monomers tend to be chemically sticky and bond together just outside the neurons and form what are called beta amyloid plaques, these clumps of lots of these monomers. These plaques can potentially get between the neurons, which can get in the way of neuron to neuron signaling. If the brain cells can't signal and relay information, then brain functions like memory can be seriously impaired. It's also thought that these plaques can start up an immune response and cause inflammation, which might damage surrounding neurons. Amyloid plaque can also deposit around blood vessels in the brain, called amyloid angiopathy, which weakens the walls of the blood vessels and increases the risk of hemorrhage, or rupture and blood loss. Here's an image of amyloid plaque on histology. These clumps are buildups of beta amyloid, and this is happening outside the cells. Another big part of Alzheimer's disease, though, are tangles, and these are actually found inside the cell, as opposed to the beta amyloid plaques. Just like other cells, neurons are held together by their cytoskeleton, which is partly made up of microtubules, these track-like structures that essentially act like a minecart, shipping nutrients and molecules along the length of the cell. A special protein called tau makes sure that these tracks don't break apart, kind of like railway ties. Although, again, it's not completely understood, it's thought that the beta amyloid plaque buildup outside the neuron initiates pathways inside the neuron that leads to activation of kinase, an enzyme that transfers phosphate groups to the tau protein. The tau protein then changes shape, stops supporting the microtubules, and clumps up with other tau proteins and gets tangled, and leads to the other characteristic finding of Alzheimer's disease, neurofibrillary tangles. Neurons with tangles in non-functioning microtubules can't signal as well, and sometimes end up undergoing apoptosis, or programmed cell death. Here's an image of histology showing these neurofibrillary tangles formed inside the neuron. As neurons die, large-scale changes start to take place in the brain. For one, the brain atrophies, or shrinks, and the gyri get narrower, which are the characteristic ridges of the brain. As those get narrower, the sulci, which are the grooves between the gyri, get wider. With atrophy, the ventricles, or fluid-filled cavities in the brain, get larger as well. So that's the pathophysiology part. But why does this happen in some people and not others? Well, Alzheimer's disease can be split into two groups, sporadic and familial. Sporadic's used to describe the late onset type where the exact cause isn't very well defined, and is probably a combination of genetic and environmental risk factors. And sporadic accounts for the vast majority of cases. With sporadic Alzheimer's, the risk increases significantly with age, affecting around 1% of people between ages 60 and 65, and 50% of people over the age of 85. In fact, a gene that's been identified as possibly contributing to an increased risk of Alzheimer's disease is the E4 allele of apolipoprotein E gene, or APOE E4. Researchers have shown that the risk of developing Alzheimer's disease increases for patients that inherit one E4 allele, and increases even more for patients who inherited two E4 alleles, one from each parent. Apolipoprotein E helps break down beta amyloid, but the E4 allele seems to be less effective than the other alleles, like the APOE E2 allele, meaning patients are more likely to develop beta amyloid plaques. Familial Alzheimer's disease, on the other hand, is used to describe cases where some dominant gene was inherited that speeds up the progression of the disease. So sometimes familial Alzheimer's disease is referred to as early onset Alzheimer's. Familial accounts for about 5 to 10% of cases, and can be caused by several gene mutations. First, mutations in the PSEN1 or PSEN2 genes on chromosome 14 or chromosome 1, respectively, have been linked to early onset Alzheimer's. These genes encode for presenilin 1 and presenilin 2, both protein subunits of gamma secretase. Mutations in these PSEN1 or PSEN2 genes can change the location where gamma secretase chops APP, producing different length beta amyloid molecules, which seem to be better at clumping up and forming plaques. Another known genetic cause of Alzheimer's is trisomy 21, or Down syndrome, which involves an extra copy of chromosome 21. It turns out that the gene responsible for producing APP is located on chromosome 21, which means that people with Down syndrome have an extra APP gene, and presumably increased expression of APP, potentially increasing the amount of amyloid plaque buildup. For this reason, familial Alzheimer's disease often progresses by age 40 in patients with Down syndrome. Symptoms of Alzheimer's disease worsens as plaques and tangles build up, and damage to the neurons accumulates. In the early stages, symptoms might not even be detectable. As it progresses, though, patients lose short-term memory, like, for example, they might not be able to remember what they had for breakfast that morning. They then progress to loss of motor skills, making things like eating difficult without help. Also, language becomes affected, making it more difficult to communicate. Eventually, they lose long-term memory, like forgetting the name of their spouse or even that they're married in the first place. And they progressively become more disoriented, which can be dangerous because they might wander from home and get lost. In the late stages, they become bedridden, and the most common cause of death is actually infection, like pneumonia. Diagnosis of Alzheimer's disease is really tough because the only way to definitively show that a person had Alzheimer's is by performing a brain biopsy after autopsy. Usually, a clinician will therefore make a diagnosis after excluding other causes of dementia. Currently, there isn't any cure for Alzheimer's disease. Some medications exist, but the benefits are small, and there haven't been any medications that clearly and definitively halt the progression of Alzheimer's. Thanks for watching.
Right? I guess now you guys know what these terminology means, like amyloid plaque, neurofibrillary tangle. So these are the proteins which leads to the degeneration part of the disease, right? So here we are talking about Alzheimer's. It's a very severe disease, as you have seen in the video that at the end there are issues such as you won't even remember, you might wander off quite far away from your home and you don't even remember where you came from. So it's a very severe disease, and uh, age related it is. So as you are aging, you start to forget things. Also, you are aging, right? So you are very fragile. So all these conditions make this disease a very active area of research, so that we can help uh, people who are suffering from this disease. It's not painful, right? The physically it is not painful, but emotionally it's very hard to manage this disease, especially for the patient as well as for the patient's family, right? So there are active, this is actually active area of research, and many advances have been made also to at least somehow detect early Alzheimer's so that we can prepare well, right? There are certain surgeries involved. So those all things are there, but still the 100% remedy is not there. So before going to our questions, there is one more small video which I want you guys to see. Again, it is related to Alzheimer's itself, but from a point of view of uh, clinical perspective, and what are the regimes and options right now available to us for curing this uh, emotionally challenging disease. Just give me a minute. Yeah, here it is. I guess you guys cannot see it. Let me reshare. Yeah, I guess now you can see it, right? It's a short one.
In healthy people, all sensations, movements, thoughts, memories, and feelings are the result of signals that pass through billions of nerve cells or neurons in the brain. Neurons constantly communicate with each other through electrical charges that travel down axons, causing the release of chemicals across tiny gaps to neighboring neurons. Other cells in the brain, such as astrocytes and microglia, clear away debris and help keep neurons healthy. In a person with Alzheimer's disease, the most basic form of dementia, toxic changes in the brain destroy this healthy balance. These changes may occur years, even decades, before the first signs of dementia. Researchers believe that this process involves two proteins, called beta amyloid and tau, which somehow become toxic to the brain. It appears that abnormal tau accumulates, eventually forming tangles inside neurons. And beta amyloid clumps into plaques, which slowly build up between neurons. As the level of amyloid reaches a tipping point, there is a rapid spread of tau throughout the brain. But tau and beta amyloid may not be the only factors involved in Alzheimer's. Other changes that affect the brain may also play a role over time. The vascular system may fail to deliver sufficient blood and nutrients to the brain. The brain may lack the glucose needed to power its activity. Chronic inflammation sets in as microglial cells fail to clear away debris, and astrocytes react to distressed microglia. Eventually, neurons lose their ability to communicate. As neurons die, the brain shrinks, beginning in the hippocampus, a part of the brain important to learning and memory. People may begin to experience memory loss, impaired decision-making, and language problems. As more neurons die throughout the brain, a person with Alzheimer's gradually loses the ability to think, remember, make decisions, and function independently. Achieving a deeper understanding of the molecular and cellular mechanisms and how they may interact is vital to the development of effective therapies. Much progress has been made in identifying various underlying factors. Advances in brain imaging allow us to see the course of plaques and tangles in the living brain. Blood and fluid biomarkers are providing insights about when the disease starts and how it progresses. More is also known about the genetic underpinnings of the disease and how they can affect particular biological pathways. These advances enable the development and testing of promising new therapies, including drugs that reduce or clear the increase of tau and amyloid proteins in the brain, therapies targeting the vascular system, glucose metabolism, and inflammation, and lifestyle interventions like exercise or diet, and behavioral approaches like social engagement that may enhance brain health. Research is moving quickly, ever closer to the day when we can delay or even prevent the devastation of dementia.
Right? So now you guys have brief knowledge or information about Alzheimer's disease. What are the reasons which till now we know that they are the reason for the disease? There can be many hidden reasons also which we are not aware of at all. So that is why it is, as I mentioned, it's an active area of research, right? So this is the article from which I have taken this image. It's a very good review. You can go through this if you are interested in Alzheimer's and you just want to know about Alzheimer's, or you know someone who is suffering from Alzheimer's. So your reading might help them also, right? Because if you understand something, you can like make situation easier for others also, right? So now, with our this generic discussion, we'll move to our questions, right, for this particular week. And this is how the plaque is seen, right? As I mentioned earlier, that I want you guys to see things how they actually are, and or how the scientists or the researchers or the doctors actually see things and diagnose and analyze. So this is not the representation of a plaque, it is actually a plaque. Okay. So we'll move to our question discussions. So our first question for today is, which of these brain reason, which one of these brain regions is associated with memory and learning? Is it hypothalamus, amygdala, visual cortex, or hippocampus? Okay. As you know the deal, I'll give you guys 1 minute for each question. So if you know the direct answer, it's good for you. If you don't know the direct answer, uh, then you can use the elimination method, first thing. Second thing, if you know the answer, good for you. But again, you should also know about the other options, that why they are not the correct ones. Okay. So your 1 minute starts now. last 10 seconds. Okay. The correct answer is hippocampus. Obviously, if you have gone through this week's lecture, you know that this was one of the predominant dedicated lecture about hippocampus and the things associated with it and the memory formation and all the case study and everything. You should go to the lecture if you haven't seen it. So this is the location and the representation of a hippocampus, right? So this is the side view of your brain, and you can clearly see that this is where your hippocampus is situated, and this is the region which is associated with memory and learning. So if you have seen the lecture, you know that if you remove this part or hippocampus, you won't be able to make any new memories, right? Or you won't be able to learn anything new. Everything will be stored in your before the removal of hippocampus. Everything will be stored there. There will be no increment of your memory and learning if you remove the hippocampus, right? So this is the representative way of and the location where the hippocampus is there. This is the real image of a brain where hippocampus is situated, and you can see that why it is called a hippocampus, right? It's because of the shape of the organ and close resemblance with the animal, right? So for today, our next question is, uh, during depolarization, which ions move inside the cell from outside? Remember at what stage we are asking this? It's the depolarization, repolarization, or other stages of action potential. Uh, Read this question, remember the action potential graph, and then try to answer. The option says, is it the sodium ion which moves inside during the depolarization, or is it potassium ion, or is it both sodium and potassium which move inside during the depolarization, or none of these ions move at all during depolarization? So your 1 minute for this particular question starts now. I'll still give 1 minute time so that people who are attending the lecture online after the live session, they can do it. The correct answer is obviously sodium ion. So if you don't remember the steps and the movement of ions and the transporters and channels which operate at different stages of the action potential, it's a very good reference diagram for that. Don't go by the exact values, because for every, if by now you guys now know that every action potential is different if we are talking about different organs and different stimuli, right? So during depolarization, voltage gated sodium channels actually gets open, and it is the time when the sodium rushes into the cell, making the cell interior change in the polarization. And the same thing happens, the opposite thing happens during the hyperpolarization, when a lot of potassium, though they still are positive ions, but during repolarization, the opposite happens. The sodium channel are closed and the potassium channels are open to maintain the polarity or the polarization effect of a neuron. Correct? So our third question for today is uh, optimum sodium ion concentration for extracellular fluid is, remember, here we are asking about the sodium concentration in the ECF, that extracellular fluid, not inside the cell. Okay. So is it 138 to 146 micromolar per liter? Is it 138 to 146 millimolar per liter? Is it 230 to 500 micromolar liter, or is it 230 to 500 millimolar per liter? Your 1 minute for this particular question starts now. And don't worry if you don't remember the exact number of the concentration, but if you are okay with the units and a closer range, it can very easily work, because it's difficult to remember the exact number. So you can remember the range of concentrations. And I will suggest you to actually have this mind map of concentration of ions, of concentration of ions both at extracellular level as well as the intracellular level, because understanding action potential and also understanding some of the transportation mechanisms, working of channels, transporters, they will actually help. This information, this knowledge will actually help you in understanding them in a very crisp and better way, if you are very clear cut about the distribution of the ions and their respective concentration. Yeah, so go ahead, give it a try. Last 20 seconds remaining for answering this question. Yeah. The correct answer is 138 to 146 millimole per liter. So if you don't remember it, I have given the link of the article from where I have taken this. And even if you're not able to find this exact concentration chart, you can go to any uh, membrane biology book, and the first chapter will definitely have this distribution of ions. And still, if you want to go deep into it, like how we calculate this and everything, so there are different core books for this channel understanding, transporters, all this. They all come under membrane biology or cell biology to some extent, for a very beginner friendly purposes. So you can clearly see that sodium is more outside the cell and around 145 millimolar range, right? And it is very less inside the cell in 12 millimolar range. And the ratio of how it is maintained, like outside to inside, with respect to outside to inside, it's 12, correct? And there will be no units, because obviously they both are in same unit. So the units will be cancelled. So the ratio will be 12. And what this give us information, that a ratio of 12, especially for sodium ion, needs to be maintained for the proper functioning of a cell or a neuron or anything, right? This distribution of ions, this concentration of ion is generic. It will change. There will be certain standard deviation associated with these number, because you cannot say that if you're looking at for example at liver, or say in stomach, or say in intestine, it will be exactly 145 millimolar, right? It will change according to the organ, but some, it will be near to this range itself. Okay, Looking at potassium, you can clearly see that it is very less outside. It's like literally only 4 millimolar in comparison to sodium, which is 145 millimolar in extracellular matrix or extracellular space. While for potassium, you can clearly see that it is 155 inside the cell, correct? And the ratio is 0.026. Here you can see the difference the ratio makes, right? For sodium, we need higher ratio. And you should remember that the ratio means outside to inside. So 12 means it's outer, the outer concentration is more. The numerator is dominant over the denominator, correct? And if it is in points or decimals, we can clearly see, which indicates that it is more inside. So we need to maintain a ratio of 0.026 for potassium for a cell to function properly, correct? And similarly, like chloride, and also in this, the calcium is not included. So you should also remember about the calcium, because these are few ions which are actually directly involved in the action potential, or say muscle contraction, or anything which is excitable, right? There are certain excitable tissues which we have covered in the last session, that there are neurons, then there are muscles. These are certain things which show excitable property, right? So for them, these ions are important, and hence you should know their concentration distribution, or at least the range, so that you can work around later. Okay. So our next question for today is this. Yeah. So which of the following cell types form the population of the dorsal pathway of the spinal cord? Is it motor neurons, interneurons, sensory neurons, or all of the above? Your 1 minute starts now. So if you have attended the lectures, these question will be pretty easy for you guys to answer. But if not, don't worry, you have the live sessions, you have the lectures recorded. You can always go back to them as per your convenience. Feel free to drop your answer in the chat box. Last 10 seconds to answer this question. Yeah, so the right answer is sensory neurons. And if you don't remember this, I have this very detailed thing about dorsal and ventral. I guess next question or the next to next question will be about the ventral pathway. So the question was that which of the following cell types form the population of the dorsal pathway, right, of the spinal cord. So the dorsal column or medial lemniscus pathway, or DCML, also known as the posterior column or PCML, is a sensory pathway of the central nervous system that conveys a sensation of fine touch, vibration, 2-point discrimination, and proprioception, that is body position or body orientation, from the skin and joints. Right? It's pretty self-explanatory. It transmits information from the body to the primary somatosensory cortex in the post-central gyrus of the parietal lobe of the brain. If you are not comfortable with these terminologies, don't worry about it. These are just parts of brain. We haven't discussed in detail about the parts of brain, but gradually we'll cover them. Okay. The pathway receives information from sensory receptors, correct, throughout the body and carries this in nerve tracts in the white matter of the dorsal column of the spinal cord of the medulla, where it is continued in the medial lemniscus on to the thalamus and relayed from there through the internal capsule and transmitted to the somatosensory cortex. The name dorsal column medial lemniscus comes from the two structures that carry the sensory information, the dorsal column of the spinal cord and the medial lemniscus in the brainstem. So in like neurology, or the scientists who study neuroscience in general, what they do when they're naming things, most of the time the name for a pathway or things contains the name of the part of the brain which it is part of, right? So for example, dorsal column medial lemniscus pathway has involvement of the dorsal column of the brain as well as the medial lemniscus part of the brain. Okay, so it is very easy in neuroscience to understand just by the name which part of the brain is involved in which pathway. Okay. Our next question is, okay, so AMPA is a, so what is AMPA? Is AMPA a glutamate receptor or GABAergic receptor, cholinergic receptor, or none of the above? So I'll obviously, again, as the rule says for this particular class, we're gonna give 1 minute for this question, though it doesn't require 1 minute, but still many people do get the right answer at the last minute. So let's hope for the best. Give a deep thinking about the question. If you know the answer, good for you. If you don't know the answer, don't worry about it. If you know the answer, you should also know why other options are wrong. And I'll repeat this again and again so that it will be easier for you guys to like attempt the final exam of this course, because you might not be aware of the right answer, but you might be aware of why the other options are not correct, right? So for that information, you should know everything about every single option. So your 1 minute starts now for this question. Ten seconds to go. Yeah. The correct answer is glutamate receptor. So what is AMPA? So the name comes from its full form, obviously, but it's an acronym. So what AMPA stands for? It is alpha amino 3 hydroxy 5 methyl 4 uh, isoxazolipropiononic acid receptor, also known as AMPA receptor or AMPAR. R stands for receptor here, or quisquelate receptor, right? So it's easier to just refer to it as a AMPA or AMPR, okay? So it is a inotropic transmembrane receptor for glutamate, right? And if you remember previous lectures, we name the neurons based upon what they are secreting or what receptors are on them, right? So it can be glutamate receptor, which has glutamate receptor on it. So it's a glutanergic or glutamate receptor. If it secretes choline or acetylcholine, it will be cholinergic receptor, right? So AMPA is a ionotropic transmembrane receptor for glutamate that mediates fast synaptic transmission in the central nervous system, right? It has been traditionally classified as a non-NMDA type receptor along with the kainate receptor. Its name is derived from its ability to be activated by the artificial glutamate analog AMPA. Okay. The receptor was first named the quisquelate receptor by Watkin and colleague after a naturally occurring agonist quisquelate and was only later given the label AMPA receptor after the selective agonist developed by Tage Horner and a colleague at the Royal Danish School of Pharmacy in Copenhagen. The GRIA2 encoded AMPA receptor ligand binding core was the first glutamate receptor ion channel domain to be crystallized, right? So if you guys are aware of crystallization, so X-ray crystallography or NMR, these are the techniques through which we can actually see the structure of a protein. And just look at this. This is the representative image, but this is how the crystal structure or the actual AMPA receptor looks like inside your cell. Okay, so you can see it's a transmembrane protein. So what are transmembrane proteins? Transmembrane proteins are the ones which are embedded in the membrane, right? So they will be transversing throughout the cell membrane. And in the previous week, we have discussed enormously about the cell membrane, right? Why it is there, what is the use of it, what kind of things are associated with the membrane. So one other thing was the transmembrane proteins, right? So transmembrane proteins are the ones which are transversing the membrane or the bilayer completely, correct? Both the leaflets of a membrane. So you can clearly see this is how the receptor actually look like inside a cell, not just to like how we draw lipid structure or how we just draw a cylinder or a rectangle to represent a transmembrane protein, right? So those are just the representative way of showing things so that it make things easier for us to understand. But this is the real structure, representative, but this is how the receptor looks like inside the cell. So this is the crystal structure of it. So you can clearly see that there is it's a receptor, right? So receptor means it will be on the membrane, it will be protruding out, it will be receiving ligands, and here the ligand is AMP, as you can see in the first line. This is the analog of glutamate, right? And it's a receptor for glutamate also. So it will be receiving glutamate on the receptive side, and then it will be cascading information and signaling to the inside cell. That is how receptors work. They receive signal from outside, certain changes occur in them, and then it leads to other biochemical pathways to take part, and hence final the result will be result is generated, right? So yeah, moving on to next question. Okay, so next question is, which of the following cell types exist in ascending pathway of the spinal cord? Okay, motor neurons, interneurons, sensory neurons, or Purkinje fiber? I'll give you guys 1 minute for this question. Try to answer it. And if not, don't worry, obviously we'll discuss. And every single information is present in the lecture. So I'll recommend you guys to go and watch the lectures. Yeah, so your time starts now. Last 10 seconds. I have a very elaborative video for this. You will guys understand very clearly what is ascending pathway, ventral pathway, because it might be confusing. Obviously, there are so many terminologies and so many pathways to remember. But if you have a clear-cut image and picture of it, it's easier to understand as well as remember, right? So the correct answer is sensory neurons. And this is the image. Don't be scared. We'll fill this image one by one, that what it says. Just give me 1 minute. I'll quickly share my video with you guys.
Within the spine.
Okay. Let me share my yeah, here it is. I hope you guys can hear this and see this. So we're gonna play this. Then you will understand what exactly that image was corresponding to. Okay.
Spinal cord, there are several different tracts. Grossly, they are divided into ascending and descending tracts. On the cross section of the spinal cord, these tracts make up the white matter, named because of the whitish colour that the myelin around the axon gives. The grey matter is located centrally, comprising the ventral and dorsal horns. The ascending pathways are responsible for transporting information from the body. These are sensory tracts. We will colour these tracts in blue, and starting from the posterior column, also known as the dorsal column, we have the fasciculus cuneatus and fasciculus gracilis. These are responsible for fine touch or tactile sensation, vibration, and proprioception. Fasciculus gracilis lies medially to fasciculus cuneatus, and the difference between the pathways is that signals from the lower limb, generally below T6, travel in the fasciculus gracilis, while signals from the upper limb, generally T6 and above, travel in the fasciculus cuneatus. You can remember this with the L in gracilis, reminding you of legs or lower limbs. These tracts run ipsilaterally through the spinal cord and decussate in the medulla of the brainstem. Therefore, a spinal cord injury will lead to loss of ipsilateral fine touch, vibration, and proprioceptive sensation. Next up, we have the anterior spinothalamic and the lateral spinothalamic pathways. The anterior spinothalamic tract is responsible for crude touch and pressure, while the lateral spinothalamic tract is responsible for pain and temperature. In contrast to the dorsal column tracts, the anterolateral pathways decussate within the spinal cord and then travel up to the brain. This means that a lesion of the spinal cord will generally cause a contralateral loss of crude touch, pressure, pain, and temperature sensation. This is not a 100% rule, however, as nerves entering the anterolateral pathways typically ascend 1 or 2 vertebral levels before they cross to the other side. Both the dorsal column and anterolateral pathways are involved in transmitting conscious sensation. The next tracts, the anterior and posterior spinocerebellar tracts and the spino-olivary tract, instead carry unconscious sensation. Specifically, they are involved in transmitting proprioceptive information of the lower limbs up to the cerebellum. In addition to these pathways, there are the cuneo-cerebellar and rostral spinocerebellar tracts that exist more superiorly. These carry the proprioceptive signals of the upper limbs and neck back to the cerebellum. Now we'll take a look at the descending pathways, the pathways responsible for muscle movement. First, we have the pyramidal tracts, which are the anterior and lateral corticospinal tracts. These carry signals down from the brain involved with voluntary movement of the limbs. The lateral corticospinal tract is comprised of decussated fibers, meaning if they originated from neurons in the left cerebral cortex, they will travel on the right side of the spinal cord. In contrast, the anterior corticospinal tract fibers do not decussate until they reach the spinal cord level, which they innervate. This then leaves the 4 extrapyramidal tracts. First is the rubrospinal tract, providing contralateral innervation involved in fine motor control. Then we have the reticulospinal tracts, which include the medial reticulospinal tract, involved in facilitation of voluntary muscle contraction and increasing muscle tone, as well as the lateral reticulospinal tract, which inhibits voluntary movement, reduces muscle tone, and plays a role in automatic breathing. Next is the vestibulospinal tract, which helps control balance and posture, and the fourth tract is then the tectospinal tract, which is involved in the coordination of head movements with vision.
Correct. So now this image is not that difficult to understand. It's a very elaborative video. Don't get overwhelmed by the words. You can easily understand them if you are like regularly reading them. For you guys, you don't need to remember each and every part of this figure. Just have a very brief knowledge and information about what comprises of ascending pathway, what comprises of descending pathway, what ascending pathway majorly involves in, and what descending pathway is majorly involved in. Okay, you don't need to remember every single thing, at least at this stage. And if in future, maybe if you guys are mastering in it or doing PhD in it, then obviously these things will become very easy for you guys to understand and remember very easily. Correct? So don't get overwhelmed. That's it. Just have a brief idea about what pathway is involved in what type of functionality. Okay. So our next question for today is, yeah, which of the following cell types are part of the ventral pathway of the spinal cord, right? So just now we have seen a video. If you have seen that video actively, you can easily answer this question. Otherwise, don't worry. The same information will work here also. Okay. So the question says, which of the following cell types are part of the ventral pathway of the spinal cord? Is it motor neurons, interneurons, sensory neurons, and Purkinje fiber, right? So your 1 minute for this particular question starts now. Yeah. Last 10 seconds remaining. So the answer is motor neuron. Obviously, the dorsal was part of sensory and the ventral is part of motor. If you don't remember, this is the figure. This video link is same as the previous one because in that particular video, both the ascending and the descending pathways are explained in a really good manner. Okay, so the link will work for both the questions. Also, you can go back to the lecture series and you can understand in a way more better with way more better understanding of these things. Yeah. So our next question for today is, in epilepsy, neurons become non-excitable, Schwann says, hyperexcitable, or none of the above. So your 1 minute for this particular question starts now. Yeah, the right answer is hyperexcitable. Obviously, I guess we know what epilepsy is, but just for more information, what exactly epilepsy is comprises or what exactly it is. So epilepsy is a disorder of the brain characterized by repeated seizures. A seizure is usually defined as a sudden alteration of behavior due to a temporary change in the electrical functioning of the brain, right? So normally the brain continuously generates tiny electrical impulses in an orderly manner, right? So the key term here is orderly pattern, correct? These impulses travel along neurons, the network of nerve cells in the brain, and throughout the whole body via chemical messengers called neurotransmitters. And I guess we are already 2 weeks into nervous system. We all know what neurotransmitters are, chemical messengers, right? Then neurons and all how signal transfers from within an neuron and in between neurons, right? So just to give you guys a little bit understanding of what epilepsy is, here's a short video about it. We'll see that. Let me share my screen. Yeah.
Epilepsy is the name of the chronic medical condition in which a child has repeated seizures. It's also called a seizure disorder. There are many types of epilepsy, ranging from benign to intense. In a healthy brain, different groups of cells, called neurons, are all firing action potentials at different times. During a seizure, firing rates are increased and groups of neurons all fire at the same time, leading to large spikes in neural activity. This activity causes physical or behavioral changes, which range from intense, full-body shaking to mild staring and lip smacking. Children's Hospital Colorado provides different types of treatment for epilepsy in children, including traditional anticonvulsant medications, dietary therapies, access to ongoing drug studies, vagal nerve stimulation, and epilepsy surgery. Surgery is an option for children who have epilepsy that is very localized and difficult to treat. The epilepsy neurology team has several tests to determine whether epilepsy surgery is an option. These tests include a prolonged video EEG done during a stay in the epilepsy monitoring unit, MRI, and functional imaging such as a PET scan, MEG scan, and others that can show areas of the brain that may be causing seizures. All this information will be discussed in a conference of several doctors who specialize in different areas of epilepsy, and the team will make recommendations about what type of surgery should be considered. If all these non-invasive tests cannot fully localize seizure onset, a procedure known as stereo EEG can more clearly identify seizure onset. In this procedure, thin wire electrodes are temporarily placed into the brain using a precise robotic guidance system. Different surgeries are available for different types of epilepsy. Open surgery to remove a lesion or even a full lobe of the brain is the most established and most common technique to stop seizures. In some cases, a minimally invasive approach done with a laser fiber can treat small, localized lesions. An entire hemisphere of the brain can be disconnected if it is severely injured and causing seizures. For patients with seizures coming from broad areas of both sides of the brain, placement of a pacemaker-like vagus nerve stimulator or disconnection of the corpus callosum can reduce seizures. Placement of an intracranial stimulator, such as the responsive neural stimulator or RNS, offers seizure relief for patients with seizures coming from critical brain areas. At Children's Colorado, we know every patient is an individual and that epilepsy has many causes and many treatments. Our team of experts will
right? So now you can see that there are n number of interventions are available for epilepsy. And all this come hand in hand with the technological advancement, because the more precise we can detect the source of the epileptic seizure, the more precisely we can make our intervention precise, right? If you don't know the localized or the point location of the initiation of the seizure, chances are you might damage other part of the brain. And brain is such a important and energy consuming as well as control center, right? So you cannot just tinker here and there in the brain, because they might lead to some other issues which you may not know until unless you are again conscious, right? So the more we know about the localized source of the seizure or the epilepsy point source, the more better treatment we can plan accordingly. Okay. So our next question for today is first model of learning, that is memory learning and how we learn, or memory and learning, right, was given by Donald Hebb, Manik Das, James J. Hickman, or Rosalind Franklin. I have given the photo of this figure, person who is involved in the model which was given, the first model for learning. And your 1 minute for this particular question starts now. Again, it's a very easy question if you have seen the video lectures. And if not, try to follow the week's lecture before attending the any live session. I'm not talking particularly about me. Try to attend the live session with some background information about what was covered in that particular week. Okay. One answer you can easily delete from this, like if you see the photo and the name. The answer is Donald Hebb. And so what is Hebb's theory, and what he actually stated is that Hebb's in 1949, right, theory postulated that the neurophysiological changes underlying learning and memory occurs in 3 stages. First, synaptic changes. Second, formation of a cell assembly. And third, formation of a phase sequence, which link the neurophysiological changes underlying learning and memory as studied by the physiologist to the study of thought and mind as conceived by cognitive psychologists. As pointed out by Third Brain, Hebb's neurophysiological postulate states that, so this is the actual statement what Hebb stated. So when an axon of a cell A is near enough to excite a cell B and repeatedly or persistently takes part in firing it, some growth process or metabolic change take place in one or both cells, such that A's efficiency as one of the cells firing B is increased, right? So you might not understand it properly. So I will recommend you to go back to the course lecture. There, this theory is very in very detail. It is explained, and you will understand what this statement, when an axon of cell A is near enough to excite a cell B, actually means. Okay. So our last question for today is, the part of the brain associated with fear is it hypothalamus, amygdala, visual cortex, or hippocampus? Your 1 minute starts for this particular question now. Associated with fear. The correct answer is amygdala, right? And this is the representative way of where the amygdala is present. You can see, right? It's this very, very small part. And in the previous question, we have seen the hippocampus, right? So it is near there. It's the amygdala. And this amygdala is the source of fear. This is the place where we sense fear, and this is the place where all the signals are processed for us to take action when we are in a fearful situation. Okay, so this is all from my side for this particular week. Thank you so much for attending, as well as for your patience. And as always, there is a question for you guys to ponder upon in this particular week. And for this week, the question is, if brain formation is guided by DNA, which is true, right? None of you will say no to it. If brain formation is guided by DNA, why all family members do not have similar thoughts, right? So we can think about this. You can think about it. If you want, we can discuss. So in this particular session, what we did basically, we understood some basic as well as intermediate level things about Alzheimer's disease. Before this, I had briefed something about how our brain functions in a very vague terms, but I have discussed about how the information is processed and what like conscious and subconscious part of a brain. So I've discussed quite a lot there. Then we have seen things about a particular neurodegenerative, or say progressive neurodegenerative disease, Alzheimer's. We've seen certain videos. Then we have seen how the plaques actually look like. Then gradually we discussed questions, what we usually do. So I hope that this session was as informative as it was for me to learn while preparing for this particular session. So hope to see you guys in the next session. The next session will be this Saturday itself. That at least till today, there are no changes in the schedule. So we'll meet on Saturday 5 to 7. Thank you so much. If you have any question, feel free to ask. And if not, you guys can move. I guess there are no questions. So we'll end the meet here. Thank you so much.